Okay. I did this morning and it didn't go well. Somewhere I'm going to pick somebody else this afternoon to do things. Big picture. We're talking about the control of force production, right? We started this last week. I know it feels like ages ago, back when we thought my Razorbacks couldn't beat Texas. Yay. On everything. Okay, so we're working our way through the many different and varied things that allow muscles to generate different amounts of force, right? So I have to pick up a 5-pound dumbbell, curl the 5-pound dumbbell, one thing happens, right? Switch and I go to 10, and now something else happens because, right, the 10-pound dumbbell weighs more than the 5-pound dumbbell. Yay, gravity. Okay, 15, right? Something else happens, and eventually I will run myself up to, I don't know if I can do a 35-pound dumbbell curl. Let's see. Yeah. I think that deserves like applause for me or something. So I already did this once this morning. I did 18 or 19 reps with a 20-pound dumbbell, and we were trying to estimate what it was. I told my bet it was about 40. So, okay. We talked about pronation last time. We talked about physiological cross-sectional area, right? So bigger muscles have more actin and myosin, all of which are going to be laid out in some manner adjacent to each other to allow us, in theory, to generate more force. Okay? So it does us no good to have a big muscle if we can't turn that muscle on and get force production out of it. And I will tell all of you that many of you, regardless of your muscle size, especially in your larger muscles like your quads, can't turn on all of that muscle. No matter what it is that you do, you can't do it. Okay? We can let you trim and let you practice. We can make it better, but you can't do that. Okay? So the second big thing in the control of force production, one is how big is your muscle? Two is going to be how much of that muscle can you engage? How many of those cross bridges can you actually engage to generate force? Okay? The primary thing that controls that we call recruitment. Okay? It's the ability of the motor cortex to signal to the alpha motor neurons in the spinal cord, have them signal down all the way down the axon across the neuromuscular junction. We're going to do excitation, contraction, coupling, right, Gabe, however we define it. We're going to get calcium release. We're going to bind to troponin. We're going to move tropomyosin. We're going to get cross bridges. Yay, we've got force. Okay. That process is called motor unit recruitment. You turn on muscle one motor unit at a time. Now, who remembers what the heck is a motor unit? Let's go back and refresh our memory. What's a motor unit? Okay, very good. One motor neuron and all the fibers to which it connects, which may be a very small number of fibers, if it's a small, slow motor unit, or it may be thousands of muscle fibers. Okay? And so that's what's going to happen there. When we do all of this, we're going to turn them on one at a time according to the size of the motor unit. Right? The size of the motor unit is determined generally by the fiber type, and the number of fibers to which that motor neuron is going to connect is determined by the size in the spinal cord of that neuron. And in general, the smaller it is, the easier it is to polarize it. And so those are the ones that get turned on first. Okay? We call this the size principle of motor unit. So when I lift 5 pounds, I use X amount of muscle in my bicep to do this. When I go to 10, I maybe haven't doubled the number of fibers, but there are more fibers that are active, and so on and so forth as I make my way up. Okay? The astounding thing is that if I do like multiple reps, and I start doing this, and I do multiple reps of the same thing, that the motor units that are turned on to allow me to generate the 10 pounds of force to lift this are going to shift. They're not always going to be the same during this continued lift. So we're going to talk about how we do something. Why is this not working? Okay. So, do I need to talk about action potentials? You guys know about action potentials? Can I skip over action potentials? A brief propagation or a brief electrical signal that's going to go across a membrane. This is how we're going to signal from the brain out to the periphery. It's how we're going to signal along the sarcolemma. We need resting membrane potentials. We need concentration gradients. I know Matt's on Zoom. We could ask biomedical engineers. If you had quantitative phys, did you guys actually have to calculate all of the things we need to get resting membrane potentials and how those things shift around? Sarah, did you guys do that? You have quant phys? Yeah, but you did do it at least. Yes? Okay. So we can alter all of these. The point of this is that to have a resting membrane potential, which you have to get an action potential, we have to have differences in concentrations of certain ions across those membranes. And that establishes these kinds of things. So we have a charge difference and we have a concentration difference. And so then when we open up the membrane and allow those ions to pass, they're going to try to move down their concentration gradients or down their ionic gradients to equalize those things. And as that happens, it will generate an action. Electricity and then it moves. Okay. So we can talk about all of these things. Really, if you want to know more about all of this, I'll be happy to give you an actual medical physiology textbook, and you can read all about these things to the heart beat's content. Okay. So I will very, very briefly mention a couple of things here when you're going to get an action potential. So what you see here in red is measuring the membrane potential in a particular cell. This could be a neuron, it could be part of the axon or a piece of an axon, it could be the sarcolemma. They're all going to look relatively similar like this. So most of these excitable cells have a resting membrane potential somewhere between about -50 and about -70 millivolts. Okay, and so what that tells us is that there is a difference in charge from inside to outside of about 70. Okay, and so what happens if this was a neuron, let's just say, is it has its resting membrane potential, and dendrites are going to pick up signals from the surrounding neurons, and they're going to start giving that signal here, and it's going to start pushing, potentially, if it's an excitatory signal, it's going to start pushing us up towards some threshold potential. This movement of things, these local potentials, are sometimes called graded potentials, where you get input and it starts moving us a little bit here. It's like being on the diving board and I'm edging out towards the edge. Okay, I mean, that's what we should be using to try to evoke our responses. So people have to jump off. We were watching people do parkour on buildings today, watching videos of that in an effort to evoke a sympathetic nervous system response to check out how some of our equipment was working, because the cold pressure test was apparently too intense. And it worked very well on Elise. She's the dude's doing first person GoPro parkour. Worked. But anyway, it's like moving yourself on the diving board. I'm inching my way out to the edge. And then when I get to a certain place, I got to jump. Okay. So you get these graded potentials, boom, 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 boom. They're going to come in, they're going to push us up. We reach some threshold potential. When that happens, you get an all or none response, because what happens is that it opens sodium and potassium channels. Because of their concentration gradients and different things, the sodium moves faster. So sodium is going to flow out. No, that's wrong. I got to think about this. Sodium's on the outside, right? No. Okay, it's been a long day. Sodium is higher in concentration on the outside, so the sodium comes in, and it makes us have a more positive environment on the inside of the cell, which drives us up this direction. Yeah, makes sense. Sodium comes in, it moves faster, right? Again, we can calculate what that is and those kinds of things. Christina?
Nothing.
Yes?
No, I'm just trying to picture it. You're good. No, I got it.
You sure?
Yeah, I got it.
I probably need to draw it. We have big sodium outside, right? Little sodium on the inside.
Okay.
Big potassium, little potassium. So here's our cell. Open this channel. It has to move like this. Okay, so sodium starts coming in. That makes more positive ions on the inside. That's going to drive our membrane potential. Boom, we get an action potential. We also have these potassium channels. The potassium moves a little bit slower than sodium. So sodium starts coming in, we polarize, sodium begins to slow down, and then potassium starts to go out. So we're losing positive charge. That's going to drive us back down. That's going to let us repolarize. Then, of course, we end up with a situation where we've sort of equaled out sodium and potassium inside and outside. So then we have to run a sodium potassium pump the sodium back out, pump the potassium back in order to fully sort of repolarize ourselves. Okay? All right. That's what's going to happen. We get two things though that I think are really, really important. One is something called hyperpolarization, where at the end we actually sort of extrude out more potassium. And so we drive our membrane potential below -70. This becomes really important because as this goes and becomes hyperpolarized, it means that we need a bigger gradient potential. We've got farther to go to get a second action potential to get to -50 or -55 at our threshold. Okay, this will become important in a few slides. I promise you. Okay, then we've got to restore the ion gradients with the sodium and potassium. These two things together, our ability to repolarize and restore the ion gradients, especially in T-tubules, is one of the things that can get messed up. Okay, the space isn't big enough to allow these things to move correctly and to run the pumps, and so we can lose our ability to send signals. Okay. The other really cool thing that the nervous system can do, and it's an entire class to talk about how and why, is especially in the spinal cord, we can adjust resting membrane potential in neurons. You can get inhibitory or excitatory inputs, and we can adjust what resting membrane potential is. So we can make, if this is a neuron, we can make it either easier by moving it towards threshold or more difficult by sort of hyperpolarizing it. We can move the resting membrane potential and make it easier and more difficult for those things to fire. And you can do that by changing posture. You could do that maybe by giving somebody caffeine or something. You can do it by having epinephrine, norepinephrine. You know, there's a whole like wide, huge sort of group of things that we can do, or we can change all that. Okay, and so we'll talk more about kind of how that might change a little bit. We can get into some of the things that go on with resistance training. There's the ability to do all of that. The nervous system is really good, and a lot of this control here in the spinal cord in neurons is where we control force production more so than if you were to look. So I'm just like, brain is up here. This is a neuron. Brain's way up here. The brain sends some signal, some kind of common big signal, and then the spinal cord adjusts this up and down to be able to decide, is the signal coming from the motor cortex going to be sufficient to get it to depolarize or not depolarize. So we'll talk a little bit more about that as we move along. Okay. Those are actions. Very, very important. This is just overlaying the potential, and then when you're getting sodium conductance and potassium conductance and how all that's going to go. So this is just moving of those ions. If you care about seeing those kinds of things. Okay. So two other things that I want you all to know about that are going to be important because it controls how we mess with force production. One is called an absolute refractory period. Okay. In the absolute refractory period, whatever piece of excitable tissue we have, an action potential has been generated, but there is a window of time during which that membrane has not yet fully repolarized or has not closed its sodium gates, as the sodium is still potentially able to go out. All right. It's usually not, but it could. But there is some absolute refractory period where these aren't closed, so you cannot get a second action potential in that tissue until we get out to some period of time following this. Okay, that's called the absolute refractory period. So it lasts until resting membrane potential has been reached and the sodium channels have closed. Okay, once this has happened, we've repolarized, we've closed our sodium channels right here. We're going to get into this hyperpolarization period. This is called a relative refractory period, whereby the potassium gates are still open. We're still running the pumps trying to get everything back to normal. And we can get a second action potential if we reopen the sodium gates, but it takes us more input. It takes a bigger input because we've hyperpolarized to drive us back to get a second. Okay. As we talk about in a few slides, how we use the number of action potentials that get fired in a given period of time to play a role in controlling force production, the absolute and relative refractory periods. Okay. All right. Talk about those two things. Talk about this. Okay. We've got neuron to muscle, right? Here's the neuromuscular junction. We've moved, we've sent these things in. I feel like we've already talked about this. Am I crazy? We did do this already, yes? Okay. All right. We talked about excitation, contraction, coupling. We talked about these things. Why is all of this in? We talked about the sliding filament theory. Okay. My apologies, guys. I don't know how we got like extra slides in there that were all things that we had covered back. Okay. So here's an illustration of motor unit recruitment patterns. Okay. So I've made this graph here where I'm showing the number of active motor units, so the number of motor units that are turned on or recruited versus muscular force. And so as you will note that as force goes up, the number of active units are going to go up. Okay. And so as this happens, let's say I need a little bit of force. I'm going to lift 5 pounds. Okay. I'm going to turn on one motor unit, and here are the fibers from that one motor unit. So this big kind of greeny yellow thing is my muscle. Here's my individual fibers. And note how generally a motor unit has fibers that are distributed relatively randomly throughout a muscle. Okay? They're not all in the same place. This is useful because if they were all in the same place, you might get, especially at low force contractions, you might get real weird contractions, right? Where all the muscle is very superficial, or all of the muscle is very deep, or it's only on the left side, not on the right side, so it's not going to contract and transfer force in a very efficient manner. So you've got a few, they're in here, they're on. Okay, that's not enough force to do whatever I want. So then I recruit a second motor unit. Now I got more, right? The second motor unit was bigger than the first. Then I do a third one. These are all type 1 fibers. Eventually, if I need more force, I would recruit a type 2A. So you see these bigger ones, they're supposed to be a different color. I don't know what has happened. My apologies. And another, and then eventually you get into the type X. Fibers get bigger. I did not have enough space to there should be more of these red dots than there were of any of the other ones in there as well. So that's how all of this goes. Size principle. Okay. Every time you do a movement, anything, your brain makes decisions in real time as to how many motor units to turn on. You ever grab something you thought was going to be heavy and it wasn't? Like a box. A lot of y'all maybe just moved recently in a box, right? You think you got any place to live?
Oh,
yeah. Okay. Are you happy about this? No more Great Dane? Okay.
I missed the Great Dane.
You missed the Great Dane. Okay. So I'm going to grab a box. I think the box is going to be heavy, and it's not. And I've generated too much force, and I fly over backwards. Okay. If I gave you something that's really heavy, and you start to try to pull and you underestimate, then sort of in real time, you begin increasing the amount that we're going to get. While you're doing something, I'm on balance. Okay. This is pretty impressive for me. I lost a lot of my balance. I'm here, doing things, right? You go to shove me, and not only do I do things like this, but I'm also going to sort of up and down my motor unit recruitment. So this happens all in real time. The brain is amazing. with its ability to control force production by changing motor unit recruitment. And we'll see about the firing rates of the products. Okay, so why do we recruit these slow or small motor units first? Because they require a smaller neural input to cause the slow motor neuron to reach its threshold and polarize. Okay, if you want to understand the math and the physiology under this, I will be happy to send you the original paper from the 50s where they discovered it, and you can read about it. But otherwise, please just take my word for it. Functionally, though, this is good for two reasons. You recruit the slow motor units first. They're the more fatigue resistant ones. Can you imagine if you had to walk or just stand or hold your posture with something, and the ones you recruited to do low force contractions were type 2X fibers that had about 2 good contractions in them, and then they were tired. You'd never be able to stand. You'd never be able to sit up, okay? Because they would just be fatigued all the time. So this way, when we do low force things like controlling posture or walking or things, we're using mostly our more fatigue resistant muscle fibers. That's good. Okay. Also, because they are small, I can add motor units. I've got one motor unit on, I turn on the second, and then a third, and a fourth. Because there's not that many fibers in them, when I recruit a new one, I do not get a big jump in force production. I get a little bit of force, but not a huge amount of force. And so I can very easily, or much more easily, gradate in a very precise manner the amount of force that I'm getting, okay, by using the small ones first, rather than kind of turning on the great big ones until the very end. And that gives us fine motor control, so that you can write and not grind your stylus into your laptop or into your tablet or something, okay? So that you can do things like paint the Sistine Chapel ceiling, whatever, or play the violin, all of these things that I can't do, right? I'm fascinated by art and music because I have no talent in either of those kinds of things. So I'm just like, this is amazing. So but that's what it does. It gives us fine control and fine gradations. Okay? And you can see in the gradations as we age, or if you have Parkinson's or MS or some kind of neuromuscular disorder, you can see what happens when we lose some of that ability, and it becomes more difficult for us to do things that have time. Okay? All right. I don't think we talked about this the other day. So right now is as good a time as we're going to talk about something called the active state. Okay? So the active state is the amount of time that calcium, and when we initiated an action potential, we crossed the neuromuscular junction, we're going through the T tubules, that calcium is out, okay? So imagine, okay, here's my muscle fiber, okay? And remember, I have no arteries, so please don't laugh. I'm going to draw the sarcoplasmic reticulum just like this. It's not sort of three-dimensional representative, okay? Here on that, I'm going to have a ryanodine receptor, okay? So when I give excitation contraction coupling, I'm going to have calcium that flows out, okay? So now I have more calcium out inside of this cell, inside my muscle cell, so that it can interact with the cross bridges, okay? So from the time that an action potential, so here's membrane potential. So from the time that the membrane potential goes, right, it fires, there's a little bit of lag, then we're going to get calcium release in there. You release a bunch of calcium, but then note that very, very quickly, all of this calcium is going to be moved back into the sarcoplasmic reticulum. Okay? It's moved back in through an enzyme called SERCA, where the calcium makes a PAs. Okay? Think of it like a big vacuum cleaner. It goes out and then it just sucks it all back in. Okay? The amount of time that calcium is outside of the sarcoplasmic reticulum at elevated levels, this amount of time is called the active state. Okay? The longer the active state, the more force we're going to get. Okay? Because calcium needs to be outside of the sarcoplasmic reticulum in order for us to be cross-bridge formation. So if this gets longer, you're going to get more force. In an animal model, I can give individual muscle fibers caffeine, and I make the active state longer, and I can get more force out of it. Okay? I'd have to give you somewhere between, depending upon how much you weigh, between 100 and 150 cups of coffee to give you the equivalent amount of caffeine that it takes for that to work in animal models. Okay? We give you guys micromolar doses. This works on that. So don't go thinking you can just eat a bunch of caffeine to have this happen. But we can do all of this. Okay? The active state matters. The calcium and sort of what happens to the calcium as we get successive action potentials matters in how much force production we're going to get, because we can change the frequency of action potentials that are coming into the muscle, and it will lengthen or it will shorten or lengthen the active state. All right, I'm going to show you guys what that looks like. So before we get into what happens when we get multiple action potentials in succession, I want to show you guys some things about individual muscle fibers when we send one action potential to that muscle. Okay, and so we can measure these things. They're called contractile properties of individual muscle fibers or of a whole muscle. It just depends upon what we do. I can artificially give one action potential to, say, a muscle fiber that I've taken out of a rat, or I can put electrodes on your leg or your arm or your face or whatever we want, and I can give you one action potential. And we can measure these kinds of things, and it will tell us a whole host of things. All right? What you guys are going to see here is this little graph. This is called twitch force. Okay? So when I give somebody one action potential, and we look at the force we get from one action potential, that's called a twitch. Okay? That's called a twitch. And so when you do a twitch, if we measure the time from when force begins here up to the peak, okay, that is called measuring time to peak tension. Okay? This identifies cross-bridge speed. So this will tell us a little bit of something about myosin ATPase speed. Okay? Here, time to peak tension. All right? So it's going to go up. So look at this. Here we have an eye muscle. Time to peak tension there to there is much faster than, say, if we were to look down here and they've drawn it wrong. This is supposed to be in the soles. They've graphed it to the gas drop, but here's the gas drop. Okay. So this one goes faster than this one. That would tell us, in the same person, that would tell us this has more fast switch fibers. Rise time is relatively similar to time to peak tension, but it is in essence measuring the front side slope of all of this. So it's 20 to 80% of max force. We get on this part where it's relatively linear with the calculated slope. It's going to tell us something very similar to time to peak tension. It tells us something about PA speed. If we look on the other side, we look on this sort of relaxation rate. So you get an action potential, you get force, and then the force goes back down. If we measure from peak halfway down, we get something called half relaxation time. Half relaxation time is a measure that reflects the removal of calcium. This reflects and tells us about circus speed, okay? The speed of the calcium ATPase. And again, you will note that what normally happens is if I have a fast time to peak tension, I also tend to have a shorter or faster half relaxation time than if I'm out here. So see, this is more of a slow twitch muscle, slower on the uptake, slower on the relaxation versus the fast twitch muscle, where I'm more up, faster up, and faster down. Okay? All of these things are things that any of you guys can measure in sort of anybody's laboratory in person. If you have a force transducer and you have an electrical stimulator, you can measure this muscle. Okay, any superficial muscle in the body, you can measure it in isolated animal preps. You can take the animal and you can open up their leg and stimulate the nerve or something. You can do it that way, or you can pull the fibers out. get a certain amount of information. These parameters are going to change in a very predictable manner based upon fiber type. They're also going to change in very predictable manners during exercise, during fatigue, in sort of other states. So you can learn quite a bit about what's going on inside the muscle just by looking at things on force curves. Okay. All right. Now the trickier part. Okay. Now the trickier part. And I struggled with this. It was hard for me when I was a student, and I feel like it's hard for me maybe sometimes to try to explain it to you guys in a way that makes very good sense. Okay. What we're going to talk about now is something called the force frequency relationship. So you've recruited a motor unit. It's on. Okay. It's on. We flipped the light switch in the room. What we're going to control and talk about now is that just because it's on, okay, muscles are like dimmer, they have like a dimmer switch. So it can be on, and it can be on a little bit, or it can be on a lot. Okay, so you're going to get a little bit of light from the muscle or a lot of light from the muscle. Okay, so one motor unit can get turned on with one twitch, one action potential. Okay. So the idea of firing frequency is our way of quantifying the number of action potentials in a given time period that are going to our motor. To make the math easy, excuse me, just assume that when I say that a twitch is one action potential, it occurs at 1 hertz. That means there's one action potential per second, okay? It's really not per second, but let's just, that makes the math kind of seem easy. If you're at 5 hertz, that means 5 action potentials per second, and so on and so forth, right? So here's the twitch, okay? We apply the action potential. There's a little bit of a lag because we got to get calcium release, and it has to interact with troponin and tropomyosin with the force, and then it relaxes back down, okay? There's a certain sort of set amount of time that this requires in mostly sort of microseconds, okay? In this graph here, I got one action potential. I got all the way back down and I relaxed, and then here I sent a second action potential. And it starts off and it looks exactly the same, okay? One of the things that happens though, if we send multiple action potentials relatively close to each other in a given period of time, so as we increase the firing rate, what's inevitably going to happen is that I've gotten some force production, I've gotten some calcium release, and then I'm starting to relax, and then here comes a second action potential, okay? That second action potential, there's still some calcium remaining in the intracellular space. Here comes a second action potential that tells the sarcoplasmic reticulum, oops, release some more calcium, okay? And so now I have whatever was left from the first one plus more from that second action potential. So now I get more force than what I had before. And then as this one starts to relax, here comes another one, and I get the same, I get even more force, okay? This process is called summation. It's the progressive increase in force production due to an increase in the release of calcium, because we are getting action potentials so close to each other that we cannot fully relax the muscle. We cannot fully resequester the calcium in between the sort of action potentials around. Does that make sense? Maybe that.
You don't get this. It's not during the whenever you can't send another one because it's hyperpolarized.
Right.
It's after that, though.
It's either after the fact that, yeah. So you've gotten another action potential. So we are at least outside of the absolute refractory period. Mm-hmm. We can't necessarily say, although at this low frequency, we probably could make an assumption that we're not even in the relative refractory period. When we get out here, we're getting a bunch really quickly. Some of them are probably in the relative refractory period, but you're just able, the motor cortex is able, it's motivated, quote unquote, enough to fire all of them at once. So here, we're outside of the absolute refractory period, okay? And so now we're going to get another.
Drew, did motor units ever, like, can fibers be innervated by different motor neurons, or is it only one motor neuron to one fiber?
It's only one motor neuron to each fiber. So the fiber is not going to get confused in some ways by like one motor neuron is telling it one thing and another one is telling it something else. And that's one of the reasons that that's the anatomical setup. So that's a good question. All right. So if you're a tactile person, think of this as tap, tap, tap. And at some point I start going faster, 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 faster. The same thing happens with pain. It's called wind up rather than summation. But like I poke you with the needle when it hurts. Then I go poke, poke, and it hurts more than sort of what the first one was. It actually begins to become more and more painful. And then I go poke, 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 and it gets a little bit more. Okay. But eventually what happens is I start sending action potential so quickly, the firing rate gets to be high enough that what happens is that you stop relaxing in between action potentials. And then what happens is that calcium is out, boom, 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 boom. And we get to a place where we've just ejected all the calcium out of the sarcoplasmic reticulum. And there's so many firings so quickly on top of each other that basically there's not time to ever, we never really shut it off long enough for the calcium ATPase to resequester the calcium. Okay? This is called a complete or a fused tetanus or a tetanic contraction. Okay? It feels, and I should have brought the freaking tension. Maybe I'll do that on Wednesday. Okay? But this feels like, when I give this to you artificially, this just is looks and feels like a regular contraction. When we're doing these things, when you are not yet reached a complete tetanus, where there is still some level of summation going, it's called an incomplete tetanus or a non-fused tetanus. Great.
So how do the relationship curves with like the different missile actions? Like would sprinting be like incomplete tetanus, or does it like how is action potential?
Oh, great. Oh, great. We're going to come back to that question in a minute. Okay. It's a good question. So, in human muscle, okay, most of the time we are able to reach a complete fused tetanus, okay, somewhere between a firing rate of maybe 8 to 10 to about 30 hertz. Okay? The reason that this is the case is because most human muscle is mixed. It's got both slow twitch and fast twitch fibers. Okay? So if the calcium ATPase works faster in fast twitch muscle, that means that it can relax faster here. So what happens is people that have more fast twitch muscles or in your fast twitch muscle fibers, it takes a higher rate of action potential to reach a complete tetanus. And so in super slow twitch muscles like the diaphragm, you may get a fused tetanus at like 5 or 6 hertz. Whereas if you've got a person that's a really, really good sprinter and you look at their quads, it may be 50 or 60 hertz. Fast twitch. Okay. Yes, we can use, and I'll show you guys what a force frequency curve looks like in a minute. Yes, we can use the slope of the curve to try to guesstimate fiber type in people without having to take a biopsy and some other kinds of things. Okay. So this is the idea of the force frequency relationship. So think of it this way. Okay. The frequency is the idea of what we sometimes call neural rate code. So each line here represents one individual action potential. What you guys see here is these are spike potentials measured from needle EMG. So I take this little fine wire needle. This is most of this through time is done in the FDI muscle right here, the muscle that makes your finger do this. So I jam the electrode in there, and I'm recording from one motor unit. Okay. And what you will notice here, they're lifting 2 grams, And you will see here's the number of action. Each spike is an action potential, not that many. Here's the same motor unit at 5 grams, so they've become more frequent. At 10 grams, they're a little bit more frequent. At 20, they're even more so. But at some point out here, this will, no matter how much more force I'm trying to get, this will stop going up. I will peak this out. Basically. So what you can do is we know what the time scale is on this. You can do the division and you can say, okay, this is at 3 hertz and this is 10 hertz and that's 18 hertz and this is 35 hertz or whatever. You can calculate these things out. Okay. You have to have needle electrodes to be able to, for the most part, to pull this out. You can't get this very easily from normal surface. Okay. So here is, and this is a curve that has been drawn from whole skeletal muscle, from a quad muscle in a person. Okay. And so what you will note here is the y-axis is torque production. So torque is muscle force multiplied by the moment arm, in this instance at the knee. And so there's some maximal torque that we're going to get here. They've just expressed the other torque relative to that. This is an easy way. You will note that we have some error bars on this. This is a curve that was kind of an average curve built across multiple people. And since everybody has a different sort of maximal torque capacity, if we express everything relative to that, we can create these curves that would be relatively similar for people. The x-axis is the artificial stimulus frequency. So this is me. How many action potentials am I sending from the stimulator to your muscle in a given second? So we start off here at about 5 hertz, and then we go to 10 and 20 and 25 and 30 and 35 and 40 and move all the way up 170. So you'll note how there's this kind of positively accelerating relationship amongst all of this. Okay. So you get big jumps here when at very, very low kind of frequencies. You go from like 5 to 10, and you get a lot of an increase in force. Let's get up here and you get a lot. But out here where it plateaus off, this is your fused complete tennis. Every person is going to have their own unique kind of plateau frequency out here. This kind of top end. Everybody's curves just ever so slightly different. A person that's a slow twitcher would have a curve that was steeper here, and then so it would be like sort of left shifted. It would be steeper on the front end and plateau at a lower frequency. A person that's a fast twitcher is going to be shifted to the right. Slower, 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 right? And then get out of this. Okay. Now, Brady, ask the question again.
I was wondering if you could like look based off the curves and the time it takes for action to just go off. Like if you could determine the muscle action, certain muscle actions are different over this time, according to the action potential.
So the short answer is yes. The long answer is yes, but it depends. Okay. What you guys see here is my single favorite graph in all of neuromuscular physiology. Okay. Because what it shows is the combination of recruitment and firing rate within the gradation of force production. Okay. So again, this is from the FDI muscle. Also, the first author of this paper, I don't know why it's on here, is the last name is Monster. So Mike, read the Monster paper. It's great. I don't really know why that always speaks to me. So we have little fine wire electrodes that have been stuck into the FDI muscle. Then you ask a person to generate a slowly kind of ramping up amount of force. Every line represents one motor unit. Okay? So we can count all of those bad boys up, and I don't know, there's like 35 or 40 or something motor units that are given here. Every line is a motor unit. Okay? Every dot on the line represents sort of a measurement at a point in time that's going to tell us about the firing rate of that individual. So what this graph does is you can see is here's force production on the x-axis, firing rate of the individual point on that motor unit on the y-axis. So when I need a tiny amount of force, the first thing that I do is I recruit one motor unit. that motor unit is firing at 8 hertz. So it's on, and it's at a relatively low firing rate. When I need a little bit more force, when I get to here, I do not recruit a second motor unit. I just turn up the firing rate in the one that was already on. I'm brightening up the light in the room by increasing the firing rate. Okay? I need a little bit more force. I'm still going to increase that firing rate. At some point, though, I recruit a second motor unit. When the second motor unit gets turned on, if we were, and of course I don't have a change room, I can put several of these things together. Need a ruler, okay? But look, when this one gets turned on, right, it's on and it's firing at 8 hertz. My first motor unit, it's still firing, but it's up here at like 16 or 17 hertz, okay? When I need more force beyond that, I turn up the firing rate of my second one and I turn up the firing rate of my first one. Okay? Then we get out here to some place where these are getting very close to their top and we recruit a third motor. So the way that all of this works is when you need more force, you want to recruit the smallest number of motor units and have them fire at the absolute lowest rate possible in order to get the bare minimum amount of force that we need. Okay? So not all motor units are going to fire at the same rate in the middle of a contraction. Some are at lower frequencies, some are at higher frequencies. Now, at maximal force levels out here, you've recruited all the motor units you can, and you're driving all of them at sort of as high a firing rate as you can for those individual units. And so in that particular instance, at a very maximal-ish contraction, you may be able to get a relatively similar firing rate across most of them. But in general, this is what all is going on. Bare number of units, lowest possible firing rates. So when I lift this, I do 5. I may have 5 motor units on, but one of them is firing at 10 hertz, one's at 20 hertz, one's at 25 hertz, okay, so on and so forth. And I need to go to 10. Maybe I'm strong enough, I assure you I'm not, I'm strong enough that I've got the same 5 units on, and all I've done is just turn up the frequency in those same 5. It's not what's happening, I promise you, okay? And I work my way all the way up, and I get to the really heavy one where it's very close to maximal. And so now I've moved my way, and they're all firing at a relatively kind of same rate. Now imagine, you don't think about this, do you? You don't think like, ooh, I better turn on 7 motor units, and I need them to fire at this and this and this. The brain just knows, okay? It just knows. And the crazier thing is, that the signal from the brain is basically the same, one big signal from the brain, but by our ability to control the resting membrane potential in the neurons, that same signal evokes either on or off in some of them, and it evokes different firing rates out of the ones that are on. Okay? And so it's a wild concept that we can even do any of this. We never have to think about it. We never think about it. So to answer your question, Brady, can we figure out what's going on, and can that tell us something about like what we're doing in a particular movement? Yes, it can, but I can't get this information unless you're going to let me like stick 50 needles into it. And the needle EMG only works. Anybody ever done needle EMG? Seen this? No? About biomedical engineers, y'all don't do this? Never seen this? Okay. Needle EMG only works during isometric muscle contractions, because if the muscle, you're doing a concentric contraction and the muscle shortens and it changes shape, then the needles are no longer in the same electrical field that they were in like half a second ago. And so you may not be recording from the same place. So they only work during isometric contractions. And they really only work when you do these kind of ramp contractions where you go up really, really slowly. So there's a lot of problems with kind of our ability to get good information like this, Brady, during dynamic, real world sort of movements. So that's sort of, we have a fancy EMG, surface EMG thing in my lab where we can sort of do this sometimes, but it's also under isometric conditions. And it's not nearly as kind of cool. We can make some really fun graphs, but it's not really as cool as this. Okay.
So what are some reasons the first motor neuron isn't continuing to produce? Why isn't that the end? It's not like how come it's not?
Oh, so like why did it turn off here?
Yeah.
It got tired.
It just gets tired.
It's fatigued. It's a concept we call motor unit cycling. And so they held this contraction long enough, which again is one of the problems. They held the contraction long enough that at this point it just dropped off and it shut off to rest. And so in this way, you can imagine that if I'm doing a submaximal contraction where I need 20% of my maximal force, I could string a huge number of combinations of motor units at certain firing rates together to be able to let me lift that 10 pounds, right? So I do it for a little while and I'm going to go here, right? I'm going to get my 10 and then this one drops off. So I turn the thing down in a couple of them and I add another one and we adjust things in that way that lets us kind of continue to do things. There's a huge amount of actual complexity that's going on in these things. Okay. Questions about that? I'm going to make a gross assumption and generalization just because of the ways that you guys brought it. We had to vote. Who do we think the person in class is that would probably curl the least amount of weight? It's probably me. Christina, you want to try this?
I honestly have no upper body strength. So.
Perfect. Let's go. Even better. I can pick on one of my own students. This is the way that we do this. Okay.
I said anything.
All right. Christina, can you curl this 10 pounds?
Yeah.
All right, that's pretty easy. All right. Curl 25. All the way down. All right, go.
Nope.
She quit on us, didn't she?
I can do it again.
No, it's fine. We got close enough. All right.
You're
So here's what we're going to do. Okay. You all saw the assumption, right? That if we let her have it from the very, very bottom, she had a hard time doing this, right? She had a hard time. Okay, Christina, put your arm at 90 degrees. Now go to the top. Shocking, I know. 30. We assume from the bottom you can't do 30?
Yeah, you want to do it?
No. Do y'all want her to do it?
Yeah.
Yes? Okay.
Ain't happening, y'all.
Go. Okay. I could feel she wasn't trying.
I was trying to.
Okay, now go to 90 degrees.
Okay, 90.
All right, go. Want to try the 35?
Hell no.
All right, let me sit down.
I'm gonna freaking break my bicep or something.
Okay. Anybody ever watch? You guys watch like shows where people are in court? Can we all, as the lawyers in the room, can we stipulate that her bicep did not magically didn't get bigger or smaller at any point during all of this? We all stipulate that her muscle size was the same. Okay. Can we stipulate that she's all, we're going to make the assumption that she has some maximal firing rate that didn't change when she was lifting different weights and that she has some maximal ability, right, that we couldn't change to recruit, okay? All of those things being equal, how in the hell can she lift the weight from here up, but she can't move it at the bottom? 30 pounds is 30 pounds. It doesn't have to shorten as much, okay? But she couldn't make it shorten at all down there. I agree, you're right, like there's not as far to have to go, but like she couldn't move it at all. and yet she could do things there.
Because there's more, when it's partially contracted, there's more overlap between the myosin and the triple myosin and actin.
Right? I know, yeah, the things that we need, there's more overlap. Okay? Reasonable. Reasonable. What, Christina?
So biomechanics.
Biomechanics. Moment on. Okay. So more complexity. We're going to have to put it to. Okay. So what we just demonstrated is actually due to a change in the moment arm. Okay. So the moment arm is the distance from where the tendon sort of inserts down here on, I don't know, I forgot my anatomy, the radius of the arm, I wanted to shoot down here. I don't know which muscle, I don't know which bone is which. Anyway, it's down here. The distance between where it inserts and the axis of rotation at the elbow, that actually changes as you move up this way. The moment arm gets bigger when you're in the middle of the range of motion. Small, bigger, smaller again, which is why there's always that sticking point in dynamic contractions. If you can get it to a certain place, you can do the rest of the movement. Okay? Think about it. I can do the squats right, like I want to squat like this. It's a lot easier than if I got to go way down. Okay? Just happy I wore my stretchy dress pants today, so I can actually do the squat. What that's not is what Drew was mentioned. But what Drew mentioned is also real. Okay, so we're going to do this, and then I'll let you guys get out of here. What I've shown you guys here is something called the length tension relationship, or it's really the sarcomere length tension relationship. Here is a sarcomere, okay? And again, these are data taken from, this is one of the original studies that kind of isolated this in the mid-60s, is that here we have a sarcomere. We've got our thick filament and our thin filaments that are going to be here. The assumption is that this here and here is maybe a Z line or something. And so if we measure the force production, okay, from an individual sarcomere, right, if you tease these things out, what you're going to see is here's the spacing. They were calling it striation spacing, but it's just the difference basically between sort of the Z lines, all right, that we were getting on all of this. The force looks like this, okay? A very short sarcomere length, practically no force. Somewhere in the middle, maximal force at a very long sarcomere length, not as much force. Looks a lot like Christina's long muscle length, not much force. In the middle, way more force, and if she could curl up far enough, we might eventually have it back down again, okay? This is a thing that is very often confused, and it's also very often not taught correctly at the undergraduate level, okay? This is what happens on sarcomere level, okay, at the sarcomere level. You have in the sarcomere some resting sarcomere length, okay? At that length, there is some amount of overlap between actin and myosin filaments, okay? Make the assumption resting length in most people is somewhere, they're at striation spacing, somewhere between like 2 and 4. So they're right about here, okay? Note that when I do this, that pretty much all of the myosin filament has some part of an actin filament to which it can bind, which gets us more cross bridges, of course. As we spread them out, okay, as the sarcomere gets stretched and lengthened, it gets pulled apart. And look, there's not an ability, there's less, or in this instance, no actin that can be bound to the myosin. If I can't bind them, I can't get any force. And force. Weird things happen at short lengths. One, you get to a place where, because the actin filaments are crossing over each other, you can't really, you've got this one trying to get pulled that way and that side trying to get, they're being pulled in opposite directions. So weird things happen and it kind of cancels itself out. And then eventually they get so short that they bump into the Z line and they can't go any farther. So you can't continue to get force that way. Okay? This is the sarcomere length tension of the thing. Lots of people refer to it as the muscle length tension. Muscle meaning whole muscle, which is what I demonstrated for you with Christina, okay? They're not the same thing. What you see here is real, but rarely ever happens in intact human muscle. Most of you guys, no matter whatsoever you put yourself, like in your bicep or your quad or whatever, moving through a range of motion, you're almost always going to be between here and here. Okay? You're not getting so much shortening or lengthening at the sarcomere level that you're rarely ever going to get off of kind of this middle portion, this optimal length portion of the sarcomere length tension. Okay? But this is a real thing. If you could stretch your sarcomeres out or shorten all of them, it will, despite your muscle being the same size, despite the same firing rate, despite the same motor unit improvement, it will get you different amounts of force production. We'll talk about a little bit more about why what we did with Christina looks and did the way it did. Thanks, guys. Now, everybody go. We're going to do top half curls, right? Because we're stronger already. We're going to go do some quarter squats today.